Alright, we're talking about enzymes in this video, and we're just going to start off with a very basic definition of an enzyme. Substrate. The word substrate is going to be key to understand what the actual enzyme is. A substrate is going to be anything that we call, or is going to be what we call anything that goes into an enzyme. Now the substrate will always have a certain shape, and the enzyme will match perfectly with that shape. So this is the enzyme, and the enzyme is going to be a protein structure that exists inside the cell. Now the active site is going to be the cut into the enzyme, kind of like a lock and a key method. The key will be the substrate and will fit exactly perfectly into the lock of the enzyme. Now a substrate is going to be the beginning parts of any chemical reaction. Just like photosynthesis, just like cellular respiration, even digestion, anything. Those beginning reactants are going to be substrates. And then the enzyme is going to bind to those substrates and speed up the rate of those chemical reactions. So a very loose definition of an enzyme is going to be a protein body that works in the cell to speed up reactions. And we're going to look into a little bit more about how it actually goes about doing that. So we're going to start off with some key vocab terms. A chemical reaction, which we should know, is going to be the process that changes one set of chemicals into another set of chemicals. The reactants are going to be on the left side of the equations, the compounds that you put into the chemical reaction. So just like the example I did a couple weeks ago in class, where I said the math equation 2 plus 2 equals 4, 2 plus 2, both of those 2's are reactants. The products are the compounds that are made by the reaction. So in that same example, 2 plus 2 equals 4, 4 would be the product. So if I was to label this chemical equation, which you should recognize as cellular respiration, I would put the glucose and the oxygen, I would label them reactants, right? They're on the left side, they're what go into the chemical reaction. And then the carbon, the water, and the ATP, those are all products of the reaction. Right? And then here's a new term that we probably haven't heard before, but activation energy. And that's going to be the amount of energy that's needed to start a chemical reaction. Now you should be familiar with the amount of energy in a molecule that's stored between the bonds. So when you have an element such as O2, that's two oxygen molecules binded together. And they're going to be binded together by sharing the electronic forces of those two electrons with the protons of each element. Now those bonds that bind those two oxygen molecules together are going to need energy to be broken. So that's why we have activation energy. We need some basic level of entry energy to start that chemical reaction. Then you have a catalyst. Now a catalyst is going to be anything that allows a reaction to occur more quickly by lowering the amount of activation energy. So let's say I was doing this chemical reaction here, and I needed something to break the glucose. So in order to break the glucose, I'm going to need to build up a certain amount of energy by breaking all of those bonds, and then once I get to a certain level, then I can start processing the chemical reaction, right? But it's going to take me a long time if I need to build up a large amount of activation energy. A catalyst is going to lower the amount of activation energy, and it kind of helps the reaction go. So it makes it go a lot more quickly because you don't need as much activation energy. An enzyme. So this is the whole purpose of this video. So an enzyme is going to be a type of a catalyst that works in living things. Sometimes the enzyme will be there to break things down, and other times it's going to be there to build things up. So it can work in both types of reactions, right? You can either come out with a bigger product, or you can break down one reactant into two smaller products. It will work both ways. So an enzyme, if you're looking at this graph, much like the idea I just talked about activation energy, let's imagine you have a chemical reaction where you have A plus B is going to go into AB. So if this red line is going to be the reaction without an enzyme, A plus B without an enzyme is going to take a long time to build up the necessary energy in order for the reaction to go through. When you have an enzyme present, that catalyst that speeds up the chemical reaction by lowering this activation energy, that reaction is going to take a lot less time. So enzymes are very beneficial when you need to have a reaction processing very quickly. So the one without the enzyme doesn't use as much energy. Or excuse me, let me rephrase that. The one with the enzyme doesn't use as much energy. And we can see that here. The blue line, the one that represents with an enzyme, does not use as much energy. So there's not as much energy needed to start the reaction. The substrate in one of these chemical reactions is going to be the substance that the enzyme acts on or changes. And that word will make more sense once we get down here. The active site, right? We should remember this from the very first picture. That's going to be the part of the enzyme that attaches to the substrate. So this A, structure A right here, will be the whole structure, will be the enzyme. Now this whole little cutout right here is going to be the active site, right? B is this active site. That's where these substrate molecules are going to bind. Now notice these substrate molecules fit perfectly into this one active site of the enzyme. Then D is going to be the enzyme again. So this is going to be a chemical reaction, right? The substrates bind into the enzyme, and then under the chemical reaction, they're going to form a E product. So here we have two smaller substrates forming into a larger product. Properties of enzymes. So these are also going to be very important. Enzymes are specific, meaning that they only work with one type of substrate. So you have enzymes that break apart carbohydrates, you have enzymes that specific job is to break apart proteins, but they're going to be very exclusive in what they do. Enzymes are also reusable. So once this enzyme goes through this reaction, this enzyme here at D can go through and do it again, right? You don't need to make a new enzyme every single time. Enzymes are going to be affected by pH, which you should be kind of familiar with. We're going to go into it later on in the unit. But how acidic or basic a solution is, enzymes can be affected by that pH or by how hot or cold that solution is, right? So if you have a high temperature, you can damage the enzyme. So their active site, right, or shape will change. So they'll no longer fit with the substrate. So you see, again, how this active site is perfectly matched for these substrates. If I put this in a very, very hot solution, I could do what we refer to as denature the enzyme. In a hot solution would change the shape of this active site, and it would not match these substrates any longer. So an enzymatic reaction is just going to be a reaction that uses an enzyme. So here's the enzyme, here's the active site, this nice cutout. You have one substrate going in, and then it's going to break out, and then at the end you're going to have two smaller products, just like the cellular respiration reaction. The same can be done the opposite direction. So an enzyme can be used to bind together two smaller substrates into a larger product, right? So you can do both. You can either break stuff down or build a larger enzyme, or build a larger product. And here again is another look at this graph. Now again, the blue line represents with the enzyme. So this reaction is going along. Let's imagine you have your reactants, right? Your 2 plus 2. That 2 plus 2, that's going to need energy. So if you don't have your enzyme, that needs a lot of energy to start the reaction. So that amount of time that you need to build up energy takes time. Sometimes in our body, we want our chemical reactions to go fast because we need those products very quickly. So that's why we have enzymes around, and they speed up the reaction. So these enzymes are going to lower that activation energy and make it so that this reaction takes a lot shorter of a time. Alright, so feel free to watch the video again. This is, especially this graph right here, this is going to be very important to understand. And I would say the very most important thing for you to take away is obviously the definition and the properties of the enzyme, but you need to be able to comprehend in your own words how this activation energy, the lowering of it, shortens the amount of reaction, and how the enzyme actually speeds up those reactions by lowering that activation energy. So if you have any questions, please bring them in the class. Write them down right now. That way you don't forget. and I'll see you guys tomorrow.